Both RNA and DNA extractions require a process of cellular lysis and washes with different chemicals, with a final stage of precipitation, though special considerations need to be made. For the DNA extraction, three main steps are going to be made. The first one is the breaking cells step, then a lipid protein and RNA removal, and a DNA precipitation. For the RNA extraction, four main steps need to be made, including cell disruption with guanidinium thiocyanide and reducing agent, an activation of contaminant proteins and breaking the disulfide bonds, the phenylchloroform isoamyl acid addition, and the washing with ethanol. For the purification of the sample, different enzymes need to be added. For example, for DNA, proteinase K and RNase treatments need to be made. For RNA extraction in general, DNase treatment needs to be made. To prevent the degradation of the samples, for DNA, EDTA solutions need to be added, and for the RNA extraction, a critical step is the stabilization of the RNA molecules, once obtained as they are easily degraded. The most common method is known as GIPS. Regarding the pH, the DNA extractions need to be made at a pH 8 for phenylchloroform solutions, while RNA extractions is with a pH of 4.8. If a DNA molecule is intended to be obtained from an RNA extraction, a reverse transcription step is needed. There are different protocols according to the type of microorganism or cell from which DNA is going to be extracted. The main differences are observed in the cellular lysis step. For lactobacillus, a gram-positive bacteria, lytic enzymes should be added in order to disrupt the multilayer membrane cell wall. For Arabidopsis thaliana, differs from the most modern and generic techniques used for animal tissues due to the cellular structure of plant material versus the animal tissue. And for saliva, a special procedure should be made in order to avoid the pollution of the sample with the possible other food residues contained in the saliva. To avoid the pollution, the PBS and blue origin chemicals help to separate the cells from the other components present in the mouth and help to perform the cellular lysis. For the protein and cellular residues elimination, some common steps such as protein precipitation solutions, RNA solutions, and proteinase K addition are made. For the precipitation and DNA centrifugation, they are common steps as the material that is going to be precipitated, it's already DNA, and it's common for all these organisms and cells.
For the work with commercial kits, it is necessary to extract the DNA from the matrix. The process consists in different phases of extraction and purification. The first step is the lysis of the cell, in which there are necessary compounds as salts to break the structure of macromolecules and the SDS detergent to eliminate the cell's membrane. Then, the degradation of the proteins interfering the sample of DNA with a protease enzyme. For the purification, the first part is the precipitation of DNA. The DNA is insoluble in alcohol, so it is precipitated in cold ethanol and recovered by centrifugation. The supernatant alcohol takes the rest of the salts away. Wash the pellet with alcohol and centrifuge again. For the purification, the first part is the precipitation of DNA. The DNA is insoluble in alcohol, so it is precipitated in cold ethanol and recovered by centrifugation. The supernatant alcohol takes the rest of the salts away. Wash the pellet with alcohol and centrifuge again. Spectrophotometry. The concentration of a molecule in solution depends on the absorbed light by the molecules in solution. DNA absorbs UV light at 260 nanometers, and it allows to determine the concentration of this nucleic acid. Spectrofluorometric DNA quantification. Ethidium bromide is useful for this quantification. It permits a great sensitive fluorometric measurement. The best results are reported with 5 micrograms over milliliter and using an excitation of 546 nanometers. Agarose gel electrophoresis is a way to quickly estimate DNA concentration. A sample of the isolated DNA is loaded into a well of the agarose gel and then exposed to an electric field. Since small DNA fragments migrate faster, the DNA is separated by size. Slot blot. This method is based on the specific hybridization of a 40 base pair probe that binds to the human alpha satellite locus. Using this technique, DNA is quantified by visual comparison of sample band intensities with band intensity of standards, produced from unknown amounts of DNA.
The results in this practice were the concentration and purity of RNA and DNA obtained respectively from bacteria E. coli and a sample of blood. This was possible with the usage of a spectrophotometer, which is an equipment that can make a quantitative measurement of the reflection of an analyte as a function of wavelength. For the RNA, we obtained a concentration of 5.42 micrograms per milliliter and a purity of 1.14. On the other hand, the DNA concentration we obtained was 3.97 micrograms per milliliter and it had a purity of 1.76. The level of purity that can be considered as acceptable is 1.7 or higher. This means that the DNA we obtain has an acceptable purity and it probably doesn't have much contamination. On the other side, the purity of the RNA was below the optimal value, so this means that it probably has a contamination of proteins or other substances. The values obtained can be compared with an electrophoresis of the gel, which was an electrophoresis of DNA and RNA in TBE buffer 1X with 8% of agarose. In the second column, it is observable how the DNA band can be seen without degradation and without any other band in the column, which means it has a good level of purity. Since it has a low level of concentration, the intensity of the color of the band is low. For the RNA, the bands can be seen with a greater intensity, and this is because of its higher concentration. However, it can be seen that the bands are degraded, and even it has a band in the charging well, so this means that it has protein contamination.
The main difference among the DNA and RNA extraction protocols rely in the nature of each macromolecule. For RNA, extra precautions should be taken for stabilizing the molecule. For the purification of the molecules, differentials in pH should be considered as the molecules have a different affinity for them. Additionally, for purification of the samples, different enzymes need to be used for eliminating the other macromolecule, being DNA or RNA. With these examples, it is clear that the differences of the chemical compositions and biochemical interactions allow the possibility of developing isolation techniques. According to the 8 to 60 over 8 to 80 index for the DNA extract, a 1.76 value was obtained. This means that the product has a good quality for working, as it enters into the acceptable range. This was confirmed with the electrophoresis gel, in which no traces of protein or RNA were observed. For the RNA sample, a 1.14 value was obtained. For RNA, the ideal value is 2, so this means the extract had a low purity. This was proven with the electrophoresis gel, in which proteins and residual DNTPs were observed in the same lane.
A ratio of approximately 1.8 is generally accepted as pure for DNA. A ratio of 2 is accepted as pure for RNA. A lower ratio indicates the presence of proteins, phenol, or other forms of contamination. As it is shown, the absorbance in both cases does not seem to be part of a pure RNA or DNA. Also, the bonds in the gel prove that the partial degradation or contamination.
As we could see in the results, a correct electrophoresis is one of a high purity level, which means it has no contamination of other components, and if it has a high level of concentration, it will be seen clearer after the electrophoresis. To achieve this, it is important to be careful in the separation and wash process, so no contamination can happen. When we make a spectrophotometry, it is important that the blank and the analyte are well prepared, are in the correct concentration, so the lectures are truthful. Finally, after the electrophoresis is made, it is important to analyze each band and each imperfection, like degradation of the bands or contamination, to know what might have gone wrong during the preparation and to avoid making the same mistakes again.